Hello, my name is Sophia Schwartz, and welcome to the STEM with Friends podcast, where I discuss a variety of science topics with interesting guests. I hope you enjoy it. To begin, we are here at Friends Academy, and we wanted to introduce our very first guest speaker, Daniel Deroulet.
Thank you so much for being with us today.
It's my pleasure. Thank you.
Yeah, so can you just walk us through your background and how you first became interested in biotech and maybe your first experiences at the Greenville School, where many of our students have attended?
Yeah, sure. So I grew up out here in Laurel Hollow. I went to Greenville, as you pointed out, from nursery through ninth grade, actually. That's back when they had the ninth grade. And from there to St. Paul's and then to Columbia. At Columbia University, I studied economics, and actually my career started in finance. I spent a decade on Wall Street in a very interesting time to be doing kind of equity research and long-short portfolio investing. That was all through the financial crisis and everything, but that's a different story. I was always interested in science, so heavy course load in boarding school and then in college in specifically the biological sciences. That's always the most interesting for me. I was close to being a bio minor at Columbia, but I was a couple labs short. And actually, over my summers, I worked at the Cold Spring Harbor DNA Learning Center on some independent projects under Scott Bronson at the time. And that was great. And it gave me a pretty good background in science. I certainly would not present myself as a PhD scientist, but I can talk the talk.
Yeah, I'm actually applying to the same program later at Cold Spring Harbor.
Oh, cool.
So, yeah.
Cool.
And were there any specific things in science that intrigued you? Like any concepts in biology that you really enjoyed learning about?
Well, sure. I mean, I think the whole story is fascinating, right? I mean, and I think hopefully we come back to this later when we talk about Pink One, but the more you spend time with biology, the more you sort of appreciate the elegance of so many of the systems. I think it's incredibly complex, but somehow the further we get into understanding things, the more there seems to be kind of this underlying order that kind of makes sense. And it's beautiful to see that as you sort of deepen your understanding of these pathways and systems, to see kind of that underlying order. I always thought that was amazing.
Yeah.
Did any teachers in your education specifically inspire you?
I mean, I had great science teachers all the way through. At Greenville, I had Peter Zaloom, who is kind of a legend over there.
Wow, he's been there for a while.
Well, yeah, he's since retired, but he was at Greenville for 50 years.
Oh, wow.
He's a legend.
Yeah. No, I had great science teachers, and then at St. Paul's, Dr. G, Teresa Gerardo, who was spectacular. So, yeah, I mean, I've always had great science teachers, but I think, those teachers helped bring clarity to difficult material. I think I always had just kind of the inherent interest in biology, though. It's not like it was something sparked in me.
What about physics or chemistry now?
I like physics. I always thought physics was pretty cool. Chemistry also, but not to the same extent as biology.
I don't like physics too much either.
Oh, really? No.
Sorry, Mr. Fish.
These are cool.
Yeah, so more about mitochondrin, right?
Mm-hmm.
Mitochondrin.
That's right.
So after your background in finance, how did you begin the company? What led you to start it?
I had actually started another company at just around the same time. That's still going today, actually. A software business called Noify, which is totally separate.
Yeah.
And I actually had some time. I had some available capacity in my own life to take on another project. And my brother-in-law was finishing his PhD at UCSF under Kayvon Shokat, and he had a very interesting, his PhD work was this very interesting work around Pink One that we'll talk about shortly. And, as you can imagine, within the family, I'd see him at Thanksgiving, Christmas. He'd update me on his work, and we'd talk about it. And it was kind of fun, and we'd go back and forth. And eventually it got to a point where when he was putting his paper together, I said, hey, have you thought about trying to commercialize this? Have you thought about trying to put together a company and take it forward? His PhD advisor, Kayvon Shokat, is a veteran of quite a few companies. So, this is not an unusual thing for a Shokat Lab discovery to be commercialized. And I said, hey, look, I can probably go get some money to put behind this. We can put together a virtual company. There are a couple of holes in the story, that'll probably make it hard to raise venture now. But hey, with, less than $1 million, we could probably check a whole bunch of boxes and take a real shot at this. And Nick and Kayvon both said, yeah, let's do it.
Yeah.
And so Mitochondrion was born.
So you used your background from economics and your business background with your company, your first company, to create Mitochondrion.
Yeah, I mean, I was kind of the finance / business guy, and Nick obviously brought the science background, and Kayvon also brought, incredible science knowledge and actually a reputation. I mean, I would be lying if I were to say that it wasn't probably a much easier, even though it's still enormously difficult, but easier for us coming out of it, having that Shokat Lab pedigree.
Do the two companies connect to each other in any
No.
ways?
No.
Completely separate?
One is a software company selling business management solutions to construction
I read
companies.
a little bit. Yeah.
But hey, it's also doing very well, which is exciting.
It's good to hear.
Yeah, thank you.
So, in the early days of Mitochondrion, how did you get it off its feet? How did you start it? Were there any specific challenges that you had to get through?
Well, sure. I mean, when you start an early stage biotech company, it's not like very, very few of them open their doors, take in a bunch of money, and then go put a molecule in the clinic. There were an enormous number of things that we had to solve for in the early days of the company. We had questions around the biology. We had questions around, the chemistry, meaning the molecules we were going to use to drug this target. We had fundamental questions about the therapeutic relevance of this target for idiopathic Parkinson's disease, which maybe we can come back to in a second. So there was a lot to do, and the question then becomes okay, you have a limited budget. What are the most essential questions? What are the ones that you really, 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 really have to answer in order for the world to stay interested in what you're working on, for you to go out and raise that next round of money? I mean, I think the funny thing about biotech, so we were talking about the software company a moment ago. I mean, the thing about building a software company is, while yes, it can be its own tough row to hoe, and it can be frustrating at difficult times too. As you grow that company, typically as you bring revenue on, that's there. It's like a flywheel effect where you're building this company, that's there. You can rely on that revenue recurring, and then you just build and build and build and build. Biotech, you raise money, and then you start dying the second you cash the check, right? So, you've got the money in the door, and you have a cash out date on the calendar, 18, 24 months out, and you got a lot to do before that money runs out, because you got to go get more.
Yeah.
And so, we had to establish what our scientific priorities were and attack them. And that's what we did.
So can you explain the science behind the drug molecule developed by Mitochondrion and how it aims to stop the progression of Parkinson's?
Sure. I mean, maybe before getting into the molecule and how it interacts with the PINK1 pathway, why don't we talk just for a brief second about Parkinson's disease,
Yes.
Familial Parkinson's versus idiopathic Parkinson's, and why the target was interesting to begin with, right? So, I mean, I think one thing I would say for your budding entrepreneur, high school science audience, is that target selection when you're looking at human disease is not a trivial thing. There are a lot of things that you could go after. 30,000 plus proteins in the body.
Yeah.
Which one you decide to focus on with your therapeutic strategy is the most fundamental choice you make in any program. And so, getting the world to believe that PINK1 was an important target for idiopathic Parkinson's and not just a familial form of the disease was, a huge hurdle for us that we had to overcome. So just to look at Parkinson's disease, so there are about 1 million people with Parkinson's in the United States. There are another 2.5 in the developed world, and many others worldwide. I don't remember what the total count is worldwide. Yeah. There's not great data in some parts of the world, but it's probably on the order of about 10 million people. Wow. It's a lot. Wow. I mean, there are there's a lot of Parkinson's disease in the world. Now, less than 10% of Parkinson's cases are linked to genetic defects. So there are genetic forms of Parkinson's disease. But the vast majority are so-called idiopathic, which means that we don't really know where they came from, right? They're just like sporadic. It arose randomly. And it turns out probably not totally randomly, but not in an easily definable genetic way. So what's really interesting about the genetic forms of Parkinson's is that a number of them tie into mitochondrial quality control pathways. PINK1 is a master regulator of mitochondrial quality control. It has kind of a co-effector in the pathway called Parkin. If you have 2 mutations in PINK1 or in Parkin, you end up with actually an early onset form of Parkinson's disease, as early as sort of 20 years old.
Really?
It can be quite early. But idiopathic Parkinson's disease, which tends to come on when you're 60, 70, et cetera, and actually the risk increases as you age, it's a disease of aging, is actually most commonly associated with a protein called alpha-synuclein. I don't know if you guys have talked at all about.
Yeah, we haven't.
That's fine.
Yeah.
But it's how Alzheimer's is associated with beta amyloid, right,
yes.
and tau. So Parkinson's, the big story is alpha-synuclein in terms of the proteinopathy. What's interesting about the PINK1 and Parkin forms of Parkin, the genetic forms we were talking about a second ago, is that they don't always, in fact, they less often have any sort of alpha-synuclein pathology. So for a long time, researchers were like, okay, sure, if you have perturbations in mitochondrial quality control, you have degeneration of dopamine neurons. So dopamine neurons are very sensitive to mitochondrial screw-ups. But maybe genetic Parkinson's and idiopathic Parkinson's are different things. You're killing the same cells, but actually the disease processes, the pathological processes are different, which is, why a lot of people looked at us like, okay, PINK1, we get it, that if you don't have functional PINK1, dopamine neurons die, but is this really going to be relevant to idiopathic Parkinson's? And that became one of the major things we had to solve for in our program. I guess I'm sorry for the monologue.
No, it's okay.
But if we come back then to, okay, why PINK1 and what did we have to do in our company to show the world that it mattered? So as I said, it's a master regulator of mitochondrial quality control. It's been known for a very long time that dopaminergic neurons are particularly susceptible to perturbations in mitochondrial quality. If you give people poisons that damage mitochondria, and there are a number of them, the dopamine neurons are the ones that die first. Mm-hmm. So, there are a lot of things that suggest this sensitivity. It turns out, and I'm kind of jumping to the end a little bit here, but it turns out that alpha-synuclein itself actually also disrupts mitochondria.
Really?
That it causes, it kind of impairs mitochondrial performance. And super interestingly, and this was work done by our collaborators, this is public, so I'm okay saying this, not only does alpha-synuclein screw up mitochondria and their function, but it also impairs mitophagy. So the way the body responds to mitochondria going haywire is it tags them, and actually that's the role of PINK1, and clears them. So it's like, okay, this one's broken. Let's remove it, and then let's signal for more downstream. Mm-hmm.
Does it signal apoptosis?
No. Actually, you're trying to avoid apoptosis in most cases, right? Because if mitochondria, get bad across the mitochondrial network, they start releasing cytochrome C, and that drives apoptosis. What you're just trying to maintain kind of the mitochondrial pool and keep it in kind of good health. And yes, if it runs away, then sure, you end up with apoptosis. But a neuron is a postmitotic cell. You mostly have all the neurons you're going to have and have had them since you were 2. Is there some neurogenesis? Probably, but mostly apoptosis is not a great strategy for postmitotic cell types. What our drug then aimed to do is, so you have PINK1, this master regulator of mitochondrial quality control. It's actually a beautifully sensitive mechanism because PINK1 is unstable and only stabilizes on damaged mitochondria and starts signaling for the clearance of that mitochondria.
What exactly stabilizes it?
Oh, this is a very long story.
Yeah.
But, you can think of PINK1 almost as like a health sensing dipstick.
Yeah. Okay.
It has a domain that goes to, it's a region very close to these channels that import things into mitochondria, and it'll go down into the mitochondrion. And if the inner membrane's polarized, I don't know how far.
Yeah.
Okay, cool. Then there's actually a protease that will cleave that tail in PINK1 and release the fragment into the cytosol, and then it gets degraded. But if the inner membrane's not polarized, it actually stabilizes and autophosphorylates, and then it achieves an catalytically active conformation, and recruits and phosphorylates parkin, and together they build long chains of ubiquitin on mitochondria, and that's recognized by the autophagic machinery of the cell. So our goal was, so there's this very sensitive mechanism that allows PINK1 to stabilize on damaged mitochondria. Let's not interfere with that. Let's make sure that we're not turning on PINK1 when it's not needed. But to the extent that it is turning on, can we kind of turn up the volume a little bit? And that's what our drugs aim to do. They were kind of, selective amplifiers of PINK1.
Was it difficult to convince others that this drug would actually be worth researching in the first place?
Oh, yeah, of course.
It was a long process.
Oh, yeah, no, it was brutally hard.
How did you do it? Where did you even start?
Yeah, so, as I said, it's you're always in this fight to get, some new piece of data that will make somebody give you a shot, right? And bear in mind, the threshold that you have to convince someone varies depending on who you're trying to convince, right? So if you're trying to say to a seed stage or a Series A stage VC, this is worth a shot, your threshold is lower than if you're going to pharma and saying, look at this beautiful program. You want to take this into the clinic, and you want to pay me a lot of money to do that. So, really, it just came down to figuring out what the essential holes were that we had to solve for each stage of the program. And it's a process. Yeah. It really is a process.
How many scientists or how many employees did you have in the company?
So, in terms of actual employees, so let me give you a super quick preface. So the biotech and biopharma industry is set up in a really interesting way. There are resources worldwide that can help you run certain aspects of your program.
Yeah.
And we took full advantage